Hi, this is Abhay Dandekar, and I'm going to talk about our project, which is enhancing rootstock-mediated systemic immunity against Pierce's disease in grafted commercial wine grape variety. Before I start, I'd like to thank the board for funding this project for years, and what we are asking for is 2 more years of funding to get the deliverable, which are these bioengineered rootstocks. So before I start, this slide essentially talks about Pierce's disease as being one of the most significant problems, and this is no mystery to all of you all. It's a disease of grape vines. It is insect transmitted. This is the insect glass-eating sharpshooter, which created a most significant problem. The bacteria is injected into the xylem, and as a consequence, it is able to travel and infect both the cyan as well as the rootstock varieties. And all 150 cyan varieties and all 30 odd rootstocks are sensitive to this disease. And what is more concerning is that this disease has been controlled with chemical pesticides. There's no genetic resistance to this disease in any grape wine, at least those that are being used in the field by growers in California. And the insect has become resistant to pesticides, and this poses a clear and present danger of containing this insect from coming northward to the pristine wine growing areas and grape wine growing areas in California. And so our idea is to engineer a rootstock and come up with a genetic solution to this problem, which is really the work that we have been doing. My lab group is listed here. I'm in the Department of Plant Sciences at UC Davis. So we were the first to demonstrate that one can engineer the root system, the rootstock, and secrete proteins that could move systemically in the plant. And these proteins disrupt the pathogen and allow it to allow innate immune responses in the plant to take care of it. The problem with Pierce's disease is that the bacteria stays in the xylem and is able to cause disease due to its virulence response. And as a consequence, it is able to suppress the immunity, and that's why it's able to infect all grape wines. And as I mentioned, all cyan varieties and all rootstocks are sensitive. Our initial work, we did a proof of concept with using Thompson seedless, where we were able to prove this concept. What we have done in this project is to engineer two important rootstocks, 1103P and 101-14, and we have engineered these so that they express the proteins, the cap proteins, and we have grafted to this. We have grafted to these to cyan variety, Chardonnay, and what we are doing, what happens in this rootstock is it expresses these cap proteins. I'm not going to go into the details about these. These proteins go up, are transmitted past the graft union, and they're secreted systemically in the plant. And when they come upon infections of the xylella, they're able to work in two ways. Directly, they will disrupt the pathogen surface, releasing plant pathogen components that then trigger the immune response to clear the pathogen. And that's how we get resistance. We now have about 69 lines or 432 different wines planted that we have been analyzing in the field, and that's the project. This picture just shows you the plantings. We had about half the plantings we planted in 2018, and the other half we planted in 2019. And this is what the plants look like. They are grown with 2 codons, and we are infecting these plants to find out which ones are resistant to show resistance uh, to the root from the rootstock. Our objectives, there are about 4 objectives. What we are doing is we are looking at analysis of disease susceptibility. We are analyzing wine health and productivity, where we harvest the grape wines and we examine and see what the yields are. We are also doing a comparative analysis. We hope to do a comparative analysis of the proteins to show that the proteins are moving up into the plant. And then in the next, as part of this proposal, we plan to also initiate the regulatory process so that we can deliver these plants to the industry so that they can do further testing. So some of this data we are already analyzed, and we are looking at all our lines, and this is the data distribution of all the wines. Each of these dots is a wine, and this is yield. This is data that we generated just last year, and you're seeing a distribution of this data, and we are looking at the top quartiles to look at the yield, and so this is the quartile. So you see a lot of the plants that have been that are grafted to these engineered rootstocks are really doing well and yielding well. We also looked at bud break, which is an example of vine health after it's being infected, and we see many plants in that wines that we can select there. And then also looking at the symptoms. In 23, and you see this, we want to have less symptoms, and so we see a lot of wines are showing less symptoms, even though they've been inoculated 2 and in many times 3 times over the last several years. We hope to do another infection this year, and then we will finish the data collection in 25, and then start the process of looking at the deregulation. These are, at the moment, these are about 11 lines that are showing really good, looking very promising. You'll see the line, they are different cap constructs, rootstock, and you're seeing how many counts of these these vines we have. This is the yield. You see they're all yielding well. This is the PD score. You can see very low PD scores. This is scores of Pierce's disease. This is the amount of symptoms, very low and below 25 quartile. And this is the bud break in the shoot. We are in the process looking at this, showing high bud break, so the wines are pretty healthy. So this is what we, just to summarize what we are planning to do in the next couple of years, this year and next year. Finishing. So these are all the lines that we have in the field. There are about 456 plants. 415 of them have been inoculated. There are 69 lines. I've shown you that 11 are already looking promising. There are many more that are looking promising, and we need to confirm that. We're going to do wine health and productivity, analyzing yield. We're going to do some analysis of the proteins to make sure what is the concentration of these proteins that are engineered into the rootstock moving up into the plant to trigger the immune response to protect the plant against Pierce's disease. And then we want to figure out these lines that we will then like to release, and we hope to do that. We want to validate the disease suppression, validate the wine health, and validate the mode of action that I talked about, where these proteins are disrupting the pathogen and stimulating the immune response. Finally, we're going to work on, this is how plants are deregulated. There's a coordinatory regulatory pathway that involves APHIS, EPA, and FDA. We are going to focus on APHIS. They have a new process which is called regulatory status review. We've got to present for the of the plants that we want to go through this process. We got to show their biological properties, show what DNA sequence we have in these plants, the trait and how it works, the mechanism of action, which is something that we need to prove to them, and we hope to do that in these couple of years. Once we get approval, that means these plants can be tested without any permits, and the industry can check these plants out, see how they work, and that's it. So that's our proposal. We hope to finish off this project, and that's what we're asking for 2 years. I thank you for your time and look forward to working with you all. Thank you.